Thank you very much, Jordi, and thank you very much to the organizers for the opportunity to explain you some of the results we have in the lab. We are interested in identifying and analyzing new targets implicated in neurodegeneration in order to design new treatment for mainly in that case Parkinson disease. So this morning I have prepared a very easy to digest presentation with the best moments of the phosphodiesterase 7. I will call it PDE to make the thing shorter. So PDE are a large family, consists of 11, I think it's not working. Okay, here. Consists of 11 subtypes, and they are largely based on their sequence homology, and they are responsible for the metabolisms of the nucleotides. They are coded by 21 identified genes, and just pay attention here. As you probably know, these nucleotides, cyclic nucleotides, are involved in a lot of events, cellular events, and in the nervous system is related to neurogenesis, apoptosis, plasticity, memory, etc. So aging and age-related disorders, Parkinson include, has a lot of impairments in many of them, if not all, these processes, suggesting that nucleotides could be compromised in patients with this illness. The PDEs are widely distributed in the human body. Most of them even has a specific location. And in particular, PDE7 is expressed in the brain. We know by previous works and by some observations made by ourselves that PDE7 is expressed in the brain in those areas related to Parkinson, like the striatum and the substantia nigra. Of special interest are the works previously published by Reyes Irizarry and the group of Guadalupe Mengot. Hi, Guadalupe. And this is why we were interested in studying the role of this PDE7 in the development of Parkinson's disease. Well, all of you know that 6-hydroxydopamine causes a time-dependent decrease in cell viability. This is why we started studying the expression of PDE7 after damage. At this time point, ¿la acabo de liar? No. And what we saw is that we could check that there is an increase in the PDE7 expression at all these time points. We confirmed these results with immunocytochemical analysis. You can see here in green the expression of PDE7, and in red is a dopaminergic marker. In Parkinson, we have neurodegeneration, but also we have neuroinflammation. So we made the same procedure in primary glial cultures. Here you can see in red microglia. Eso es que ya no. In red microglia expressing PDE7 in green during all the time points studied. We confirm again the results by Western blot. We performed the same analysis in astrocytes cultures, and we saw, here you see the astrocytes marker in red, and PDE7 in green. We see an increase in the expression of PDE7, and we confirm again the results with Western blot. We found here a difference in comparison with microglial cells, that is PDE7 is less expressed in astraglia cells in comparison with the microglial cells, and this expression starts later in comparison again with the microglial cells. So next step was to use embryonic mesencephalic cultures. Why? Because here we have neuron precursors together with the glial cells. This is why we treated these cultures with LPS to provoke neuroinflammation, and with cis-hydroxydopamine to provoke oxidative stress. Again, we saw an increase in the expression of PDE7 at all times studied. Where was exactly this expression? In which cellular type? We performed immunocytochemical analysis. Here you can see in red the dopaminergic precursors, here in these mixed cultures, expressing PDE7 at all stages, and the same results with microglia and astrocytes. Here microglia and astrocytes in red, showing an increased PDE7 expression. So what we have up to now is that in vitro we have an increased expression of PDE7 in neurons and glial cultures. This is why next step was to move to a more clinically relevant model. We have rats intracranial injected in the substantia nigra with LPS in a model of neuroinflammation and with 6-hydroxydopamine in an oxidative stress animal model. What we saw here is, as a consequence of the damage, a decrease in the number of dopaminergic neurons linked to an increase in this dopaminergic neuron of PDE7 here in green. What we saw in the substantia nigra is that these degenerating dopaminergic neurons, here labeled in red, was expressing PDE7 at the same time that pro-apoptotic markers. On the other hand, we could observe some dopaminergic neurons intact. They were not degenerating, and they didn't express PDE7 or pro-apoptotic markers. This with the two models, with the inflammatory model and with the oxidative stress model. When we studied the glial surrounding these dopaminergic cells, we got similar results. You can see here microglial cells and here astrocytes that are expressing PDE7. Again, we could see that the expression in astrocytes is increased, the expression of PDE7, but is lower in comparison with the microglial cells. And finally, we performed some transient transfections with a fragment of the human PDE7 promoter into neurons, microglial cells, and astrocytes. What we could observe is that this transfection, after damage, provoked an increased luciferase activity. So, as I told you, this is a brief summary of all the results we have regarding to PDE7. You can go to this paper that we have just published 2 weeks ago, and bear in mind that you are here, you have the last chance to go to the poster number 5 and poster number 55 to see in detail these results. So sorry, Marina is not going to be there. And so to sum up, the highlight is that PDE7 is highly expressed after oxidative stress and pro-inflammatory conditions, both in vitro and in vivo, in neurons, in microglial cells, and in astrocytes. So this is why we think that PDE7 could be necessary to start to the onset of the illness and maybe to the progression of the disease. And this is why we think that it could be a new target to design new treatments to prevent the PD progression. We are in the Institute for Biomedical Research in Madrid, so you can find us there. This is all the group. Thank you very much to Ana Pérez Castillo for your excellent mentoring, and to all of you for your attention. Thank you very much.
Questions? No questions? Just a quick one, because maybe I missed it when you were explaining that. Apparently, when you have some protected dopaminergic neurons in a particular area of the brain, together, I mean, not far away from other areas where you have degenerating neurons. Why is that happening? I mean, those areas have any specific? Sorry. So I'm going to repeat the question. So you have some areas that seem to be protected from the degeneration together. I mean, right on the side of others that are not protected, are degenerating. Yeah. So how do you explain that geographical differences?
Because in the substantia nigra we have the dopaminergic neuron are spreading. So at the beginning of the illness, the dopaminergic neurons more affected are those ones related to the pars lateralis. Otherwise, the neurons near the ventral area are not so affected. So as the neurodegeneration is going higher, is affecting the other part. So we have first the pars lateralis more affected, and then the other areas. This is why at the first stage, this is 72 hours after damage, we have just the beginning of the substantia nigra affected and not the other one.